Hi, my dear students. Welcome to my channel. Today, they are discussing the topic about 12th botany, asexual and sexual reproduction in plants, part 7. In the part 7, they are discussing the topic of post-fertilization structure and events and the endosperm. First of all, discussing the topic about post-fertilization structure and events. After fertilization, several changes take place in the floral parts up to the formation of the seed. The events after fertilization, that is endosperm, embryo development, formation of seed and fruits, are called post-fertilization changes. After fertilization, all the parts of the flower, such as bracteole, calyx, corolla, stamen, etc., except the ovary, wither away. The ovary forms the fruit. The ovary wall forms the fruit wall, that is pericarp. The style and stigma dry up. The ovule form the seed. The punicle persists in the seed. The hilum remains. The chalaza persists. The outer integument forms the test of the seed. The inner integument forms the tegument of the seed. The micropyle remains in the seed. The nucellus is used of the developing zygote. Sometimes it may remain as perisperm, as in Nelumbium. The fertilized egg developed to form the embryo. The fertilized polar nuclei develop to form the endosperm. The synergids are degenerate. The antipodals are degenerate. Next, discussing the topic about endosperm. Endosperm is a nutritive tissue for the embryo stored in the seed, and it is formed by the fusion of one male gamete with the polar nuclei. The endosperm is a nutritive tissue of the seed. It lies inside the embryo sac. It contains starch and oil. It is used by the developing embryo. The endosperm is formed by the fusion of one male gamete and two polar nuclei. Hence, the endosperm is triploid. Next one, the endospermous seed or albuminous seed. The seed containing the endosperm may persist in the seed up to the stage of seed germination. Example, castor and coconut. Another one, non-endospermous seeds or non-albuminous seeds. The seeds which does not contain endosperm. Example, peas and beans. So the endosperm is found in two groups of plant, namely angiosperm and gymnosperm. In gymnosperm, the endosperm is formed before fertilization and is haploid. The angiosperm endosperm is formed after fertilization. One male gamete fused with the two polar nuclei to form primary endosperm nucleus. The primary endosperm nucleus develops into the endosperm. The endosperm is found in the embryo sac. The endosperm is formed by the fusion of three nuclei, and this fusion is called triple fusion. So the endosperm is triploid. The endosperm is absent in few families of angiosperm. Example, Orchidaceae, Podostomaceae and Trapezae. In some seeds, the endosperm is used up by the developing embryo. The seed without endosperm are called non-endospermous seeds or non-albuminous seed. In some other seed, the endosperm may persist in the seed up to the stage of seed germination. That is called endospermous seeds or albuminous seeds. Another one, types of endosperm. The endosperm is classified into three types. Charnip in the year 1929 on the basis of their mode of development. Number 1, nuclear endosperm. Number 2, cellular endosperm. Number 3, helobial endosperm. Nuclear and cellular endosperms are found in the dicotyledon, and helobial endosperm is seen in the monocotyledons. Best one, nuclear endosperm. In nuclear endosperm, the division of the primary endosperm nucleus and its further divisions are not followed by wall formation. All the nuclei remain free in the cytoplasm of the embryo sac. Example, coccinea, capsula and arachis. The nuclear endosperm is found in dicot plant. The nuclear endosperm is also known as non-cellular endosperm. Davis in 1966 has recorded nuclear endosperm is 161 family among 288 family of angiosperm. In this type, the primary endosperm nucleus undergo mitotic division to form several thousands of free nuclei. All the nuclei remain free in the cytoplasm of the embryo sac. The nuclear divisions are not followed by wall formation. Thus, a multinucleate endosperm is found. Hence, this is a type is known as nuclear endosperm. In some plants, the nuclear endosperm become cellular at later stage. Example, Phaseolus and Acalypha. The free nuclei are pushed into the periphery of the embryo sac. A vacuole is formed in the center. The wall formation is centripetal, that is from periphery towards the center. The cellularization of the nuclear endosperm shows variation. In Acalypha, the endosperm becomes cellular completely. In Crotalaria, the wall formation occurs only in the micropellar region of the embryo sac, but the chalosal region remain free nuclear and elongate to form Haustoria. In Grevillea, the micropellar region, the endosperm becomes cellular and the free nuclear portion of the endosperm is in the form of vermiform Haustorium at the chalosal end, given by Kaushik in 1941. Nuclear endosperm Haustoria are reported in several members of the families like Cucurbitaceae and Leguminosae, etc. In coconut, the endosperm is nuclear type. Later on, it becomes cellular. The primary endosperm nucleus undergoes several free nuclear division. These free nuclei float in the fluid of the embryo sac. At a later stage, the fluid shows several cells. Each cell encloses a few nuclei. These cells with nuclei settle gradually towards the periphery of the cavity and cellular endosperm is formed. This is called coconut meat. The endosperm in coconut is partially solid with liquid in the cavity. In mature coconut, the liquid endosperm does not contain free nuclei or cells and it turns milky. In areca nut, the development of endosperm is like that of coconut, but the embryo sac cavity is small. These sac completely filled by the growth of the endosperm and it becomes very hard at a later stage. Second one, cellular endosperm. In cellular endosperm, the division of the primary endosperm nucleus and its further divisions are followed by immediate wall formation and the free nuclear stage is completely absent. Cellular endosperm is common in dicot. Example, Adaxa, Chelianthes and Scoparia. In this type, the endosperm is cellular from the early development stage. Free nuclear stage is absent. Haustoria are common in cellular endosperm. The primary endosperm nucleus divides into two daughter nuclei. The nuclear division is followed by the formation of a transverse wall. As a result, the endosperm is two-celled. Each cell undergo another transverse division. As a result, 4 cells are formed. Further division of the endosperms are in different plane. The embryo sac is filled with cellular endosperm. The Haustoria occur commonly in cellular endosperm. The Haustoria may arise at the micropellar end, at the chalosal end, or at both. In impotens and hydrocera, micropellar Haustorium is produced. In Magnolia arborata, a chalosal Haustorium is formed. The cellular endosperm is formed in the micropellar chamber. In Nemopila, both micropellar and callosal Haustoria are found. The callosal Haustoria has a lateral branch. Third one, helobial endosperm. Helobial endosperm is an intermediate type between the nuclear and cellular types, and the burst division of the primary endosperm nucleus is followed by wall formation, and further division results in the formation of free nuclei only. Helobial endosperm is more common in monocot. It occurs in the family of the order Helobiales, such as Butomaceae, Hydrocharidaceae, etc. And hence it is known as helobial endosperm. Davis 1966 analyzed the presence of helobial endosperm in 14 monocot family. The development of helobial endosperm is intermediate between nuclear and cellular type. During development, the primary endosperm nucleus moves to the callosal end of the embryo sac and divides into two nuclei, followed by wall formation. As a result, two unequal chambers are formed, namely large micropellar chamber and the small callosal chamber. In the micropellar cell, the nucleus undergo free nuclear division and become xenocytic and free nuclear. The cell wall formation occurs in the micropellar chamber at a later stage to form the endosperm tissue. The nucellus, the nucleus of the callosal chamber, remains undivided, or it may be undergo a free nuclear division and finally it become reduced. The Haustoria are produced by the endosperm and they are helpful in the absorption of food from the surrounding tissue. In helobial endosperm also Haustoria are formed. The development of lateral Haustoria is seen in Monochoria vaginalis. Other types of endosperm. Based on the nature of food material, the endosperm is various types. Starchy endosperm. In this type, starch is stored in the endosperm in the form of starch grains. Example, cereals. In cereals, the outermost layer is known as aleurone layer, which store protein, phytin, etc. Starchy endosperm is also known as farinaceous endosperm. Next one, oily endosperm. In this type, oil is stored in the endosperm. Example, resinous and coconut. Next one, corny endosperm. In this type, the endosperm is very hard due to the storage of hemicellulose in the cell. Example, boraces. Other type of endosperm, ruminate endosperm. During the endosperm development, finger-like outgrowth or rumination are produced from the inner face of the integument into the endosperm, and the endosperm is irregularly rigid and furrowed. Example, Arica catigo in Anonaceae family. Definite ingrowth or inbolding of the seed coat produces ruminate endosperm. Second one, example, Myristica, Aristolocaceae family. The irregular surface of the seed coat makes endosperm ruminate. In Passiflora, the unequal radial elongation of the layer of seed coat results in the rumination of endosperm. Another one, perisperm. The persistent nucellus in the seed is known as perisperm. Perisperm is found in the members of Piperaceae, Gingibraceae and Nimbaceae. The nucellus of the ovule is usually used to have the developing embryo and endosperm. But in the ovules of some plant, a portion of the nucellus is left behind in the seed. This persistent nucellus is called perisperm. Example, pepper, black pepper, nelumbium, etc. The perisperm is a Piper longum or Piper nigrum. The perisperm is a massive and the endosperm is much reduced. The perisperm is diploid as it is developed from the nucellus, but the endosperm is a triploid tissue. Another one, function of endosperm. It is a nutritive tissue for the developing embryo. In majority of the angiosperm, The zygote divides only after the development of endosperm. Endosperm regulates the previous mode of embryo development. Endosperm Haustoria. Another interesting feature of the endosperm is the presence of Haustoria. In the case of kilobel endosperm, the callosal chamber itself acts as a haustorial structure. In cellular and nuclear endosperm, special structures are produced towards the micropillar and callosal, both micropillar and callosal, which may be lateral direction depending on the species. They absorb nutrients from outer tissue or from ovary tissue and supplying the growing embryo. Then additional information, aileron tissue. Aileron tissue consists of a highly specialized cell of one or few layers which are around the endosperm of cereals, example barley and maize. Aileron grain contains perisome. During seed germination, cells secrete contain hydrolytic enzyme like amylases, proteases, which digest reserve food material present in the endosperm cell. Another one, coconut milk is a basic nutrient medium which induces the differentiation of the embryo, that is called embryoid, and plantlets from various plant tissue. Coconut water from the tender coconut is pre-nuclear endosperm, and white kernel part is cellular. Next one, evaluation. In Haplopappus gracilis, number of chromosome in cell of nucellus is 4. What will be the chromosome number in primary endosperm cell? Option A 8, option B 12, option C 6, option D 2. So the correct answer option C 6. Second question, ruminant endosperm is found in A. Coccus, option B. Arica, option C. Vallisneria, option D. Arachis. So the answer option B. Arica. Third question, when a diploid female plant is crossed with a tetraploid male, the ploidy of endosperm cell is a result of seed. Option A pentaploidy, option A B diploidy, option C triploidy, option D tetraploidy. So the correct answer option D tetraploidy. Fourth question, the coconut water from tender coconut represent option A endocarp, option B fleshy mesocarp, option C free nuclear proembryo, option D free nuclear endosperm. So the correct answer option D free nuclear endosperm. Fifth question, perisperm differs from endosperm in option A being capillary tissue, option B having no reserve food, option C being a diploid tissue, option D its formation by fusion of secondary nucleus with several sperms. So the correct answer option C being a diploid tissue. Then sixth question, the endosperm of angiosperm is different from gymnosperm. Do you agree? Justify your answer. So the answer, the endosperm is gymnosperm is caploid because it is found from the caploid megaspore. In angiosperm, the endosperm is triploid, a product or triple fusion. Seventh question, why does the zygote divides only after the division of primary endosperm cell? So the answer, the primary endosperm nucleus divides immediately after fertilization. It develops into an endosperm, which is a nuclear tissue and is a regulatory structure. The zygote divides only after that to form an embryo, so that is can get nourishment from the endosperm that has formed earlier. Eighth question, give example for kilobel endosperm. Ninth question, what is endosperm? Explain the type. 10th question, what are the post-fertilization changes? So this session, the discussing the topic about post-fertilization changes and the endosperm. So the next session, the discussing the topic about embryo. So you study the previous portion and this portion clearly. you will get more marks. Thank you.